Okay, so this next lecture is going to be about liver and liver metabolism. We've already talked quite a bit about the liver, and so a lot of this is going to be review, but there are going to be some new things, especially with respect to detoxing. So the liver, of course, is very important. It has many functions. The liver is the main metabolic organ that's going to filter things coming in through your gut. So anything entering through your digestive system and being absorbed, whether it's nutrients, vitamins, xenobiotics, drugs, it's going to pass through the liver, and the liver is the main filter. So it's the main filter for digestive tract. It is also going to be important for maintaining blood glucose levels, of course. It also has to process fats, carbohydrates, proteins, synthesize fats, ketone bodies, glucose, and it also takes in excess nitrogen and converts to urea. It also is going to make your cholesterol. It's going to convert excess nitrogen to urea. And so we've talked a lot about these functions of the liver, and so now we're just going to talk a little bit more about the liver itself and some important detoxing especially, and then some other aspects of the liver. So the liver histologically you already have seen, and it's going to be positioned between the circulation system and the GI tract. And the GI tract, of course, is where it will filter what comes in. And then the circulation or circulatory system is where it will release compounds. And it can be many different types of compounds, and the compounds it's going to make itself. And it will release blood proteins, it will release fats, etc. The sources of blood within the liver is going to be 20 to 25% from cardiac. And then the portal vein is what is going to deliver the nutrients and substances from the GI tract. And the hepatic artery is going to be from the general circulation back to the liver. So now the liver, this is just another drawing of the liver, and within this drawing you can see the sinusoids, and the sinusoids are expandable vascular channels. And they're going to be lined with endothelial cells, and the endothelial cells are going to be leaky. So they don't form tight junctions, etc. And so the reason your endothelial cells are leaky is that you want to have blood plasma have access to the hepatocytes, because the hepatocytes are the major filter. So they're going to filter the blood. Now, the liver is considered an exocrine organ. Since it will secrete bile. And the bile is going to go into the biliary drainage system. And then, of course, within the bile is going to help to secrete xenobiotics and substances out of the body and into the digestive tract and ultimately out and be excreted from the system. Hepatocytes are going to secrete the bile into the bile caniculus, and then the bile caniculus, of course, will go to the bile duct. You also have the common duct, and the common duct releases the bile into the actual intestine, into the duodenum. Some of the bile will be stored in, of course, the gallbladder, and this will be discharged during digestion. The liver itself is going to be covered by a capsule connective tissue, and the connective tissue is actually going to form branches and extensions throughout the liver. So now we want to talk about the different cell types in the liver. So the primary cell type is going to be the hepatocyte. And the hepatocyte is also called the hepatic parenchymal cells. And other cells are going to be non-parenchymal cells, and these are going to be linings of the cells, walls of the sinusoids primarily. And then all others are about 40% across the board. The lining of the cells are going to be, or the lining of the sinusoids are going to be comprised of your endothelial cells, your Kupffer cells, your stellate cells, and you also have pit cells that are going to be part of the lining cells. So first, let's just talk briefly about hepatocytes. Hepatocytes are what we primarily have been focusing on in biochemistry, and they are going to carry out the functions of the liver. And of course, the hepatocytes are going to be influenced a lot by hormones. And they are going to be what are called quiescent cells, and so they have a very low turnover and a long lifespan. Quiescent does not mean they're not metabolically active, it just means they don't divide much. So the liver mass has basically a constant relationship to the total body mass of adults. And so what that means is as you grow, your liver grows, etc. Now the next cell are the endothelial cells. And so these are the sinusoidal endothelial cells, which line, which basically make up the lining of the sinusoid. But they are unlike other endothelial cells and other tissues because they are leaky, so they do not form a tight membrane. And so there's no barrier in between them. And so they're going to allow for the free diffusion of small molecules, and basically up to about the particle size of kilomicrons. They have fenestrations or pores that are also going to increase the free passage, especially of blood components, into the liver. The next set of cells are our Kupffer cells. So they're shown here in the picture. You can see that they are associated or part of the lining of your sinusoids. Kupffer cells are basically tissue macrophages. So they're part of the immune system. And just because they happen to be in the liver, they have a particular name called Kupffers. So macrophages are going to be called different names depending on what tissue they reside in. So macrophages that are in the liver are called Kupffer cells. Now macrophages or Kupffer cells are going to be both endocytotic as well as phagocytic. They're going to be able to eat things. They can take up proteins that are denatured. So they are very responsible for homeostasis. So they help with homeostasis. They can remove denatured proteins. They can eat bacteria. And also they can pick up different types of proteins that can aggregate, such as immune complexes. They also remove damaged red blood cells. Even though most red blood cells will be removed by your spleen, your splenic macrophages, your macrophages in your liver can also remove damaged blood cells. We also have hepatic stellate cells or ioto cells, and these cells are usually not normally seen in a typical H&E stain. So it's really only in a disease state that they are going to be visualized because they become fat-laden, basically. There are about 5 to 20 stellate cells per 100 hepatocytes. Per 100 hepatocytes. They are going to be storage for vitamin A. So this is where they are going to be stored. They are lipid filled. Remember, vitamin A is fat soluble. They are shown in this picture as being fat-laden cells in the space of discs. They are typically activated after an injury, and they become more fat-laden when you have hypervitaminosis type A. Okay, so they're also called Ito cells as well. Next up are our pit cells. These are referred to as large granular lymphocytes. And they function as natural killer cells. And as their name predicts, they do kill cells. So they're going to be found inside the sinusoidal lumen. This is an electron micrograph of these. And they're going to adhere to endothelial cells as well as interact with Kupffer cells. So basically, pit cells, or what they really are called natural killer cells, are going to be a very important defense mechanism against potential invasions of the liver by viruses, tumor cells, etc. Okay, so the liver, as we know, is central receiving and recycling center of the body. They're going to get things that are coming in through the GI tract. They're going to get things coming from the heart. So they are really there to sense, to monitor. They also have recycling capabilities. They have distribution capabilities. They're going to make a lot of the serum proteins, release them. They're going to also be important for removing toxic compounds. So detoxification is going to be a major function as listed here for the liver. The liver is going to remove the toxic compounds that are either ingested or produced by us inside during metabolism, and it's going to make them more excretable, targeting them to get excreted into either the urine or bile. Now, the liver is going to, of course, receive nutrient-rich blood, and that's going through the circulation. And so the compounds that enter the blood from the digestive tract are also going to pass the liver on their way to other tissues. So when you take drugs or anything, all of those are going to hit the liver first. So keep in mind for detoxification that xenobiotics are those that have no nutrient value and can be potentially toxic. And so these xenobiotics can be things that are added to the food. They can have some pharmacologic properties. They can be recreational drugs, and most are going to be lipophilic. And so basically, in order to excrete the xenobiotics, they have to be converted to a substrate that can be conjugated. And the conjugation is going to allow them to be more soluble and allow them to be more easily excreted. So we have here phase 1 reactions. And phase 1 reactions will generally introduce or expose hydroxyl groups or other sites. And these can be conjugated then in phase 2 reactions. So phase 1 reactions include reduction, oxidation, hydroxylation, hydrolysis, and it's primarily going to be to add or expose OH groups. And then the phase 2 reactions are going to add negative charges to the group, such as a glycine or a sulfate. Now, sulfonation is used in the liver, and this is used to clear steroid hormones, for example. And then the sulfate, of course, is going to come from the breakdown of cysteine or methionine. Of course, as you guys know. So a lot of xenobiotics are going to contain aromatic rings, such as the benzopyrene ring in tobacco smoke. Or they can contain a heterocyclic ring, and those are in nicotine, for example, pyridoxine, and we can't degrade or recycle these components. And so the xenobiotics will again be reduced and then add negative charges, and then they will eventually get excreted. So we also have cytochrome P450s in our endoplasmic reticulum of our liver, and these are going to be very important in degrading exogenous compounds. So these can degrade and the exogenous compounds can be chemicals, potential carcinogens, as well as just simply pollutants. They can also be just food sources. They also are going to be important to break down endogenous compounds. So if you don't know the difference, exogenous means it comes from the outside. Endogenous means it's something that is made from the inside. And so these endogenous are going to be steroids. We have our prostaglandins, our retinoids. And so the flavoproteins, which are the cytochrome P450 compounds, cytochrome P450, they have the NADPH. You also have your cytochrome P450 reductase, and this is, of course, where you're moving around those electrons. And you have your cytochrome P450 which is where you get your R, which is your, in this case, we're talking about xenobiotics, so your R is going to be the xenobiotic. And then you have oxygen, and so you're going to add this OH group to your xenobiotic. And then again, this will make it able to be sulfonated or have a glycine added to it or something, and so it can be excreted. Now, again, the major role is to oxidize. The different xenobiotics. And so you have many of these enzymes. There are at least up to 150 different isozymes of P450s. And the naming of the enzymes are based on a lot of times what it was originally identified to be oxidizing or getting rid of. So in the case of, let's talk about naming just for a second because we're going to talk about many of these. In the case of CYP 2E1, you have the CYP, and this is going to denote the cytochrome P450. And then you have your 2, which is going to give you the subfamily. And then you have your E, which is going to give you ethanol. And then the 1 is a specific isozyme. Now, does it mean that the CYP2E1 only metabolizes ethanol? No. But this just means this is what it was identified, so this is how it got its name. All right, so the P450s have certain features in common. So they are all going to contain the cytochrome P450 that is going to be used to oxidize the substrate and reduce oxygen. And another thing is they are all going to have a flavin-containing reductase. That uses NADPH. Oops. And they're all going to be found on the smooth ER. And are referred to as microsomal enzymes. They're all bound to the lipid portion of the membrane, and it's thought that they're mostly bound, or they're probably all going to be bound to phosphatidylcholine. They're all going to be inducible by their substrate. So for example, the cytochrome P450 CYP2E1 levels will increase in alcoholics. So the more alcohol you drink, the more of the actual cytochrome P450 2E1 your cells will produce to metabolize the alcohol. So the presence of alcohol will induce the expression of the cytochrome P450. And they're also going to have the potential to generate free radicals. And these are as intermediates. So again, it goes back to how much of the xenobiotic is present. So if you have enough, or you have a low amount of the xenobiotic, then your P450s can metabolize them, and the free radicals will not form toxic compounds. However, if you overwhelm the system, then those free radicals can form those toxic intermediates, and they can do damage to the actual cell. Now let's talk a little bit about some of these. So we have CYP3A4, and this is about 60% of all CYP450 enzymes in the liver. And your gut also has CYP3A4, and that's about 70% of your gut P450s. So CYP3A4 is going to metabolize a lot of drugs and a lot of xenobiotics that we ingest. So if you ingest 2 drugs that are substrates for CYP3A4, this can induce competition. And so this is going to be competition for the binding sites, and this could lead to differences in the drug degradation as well as altering the levels of the blood levels of the drugs. Now, some of the things that are important is if you have something that is going to impair or need to be metabolized by the 3A4, and then you give a drug against that also needs to be metabolized by the 3A4, they can sort of counter each other. And so grapefruit is an inhibitor of CYP3A4. And so you can't take 2 drugs or you can't take a drug that needs to be metabolized by CYP3A4 and drink grapefruit juice. So a lot of times in the directions for drugs, it'll say it cannot drink grapefruit juice while on this drug, for example. And it's because the enzyme that's metabolizing the drug would have to metabolize grapefruit juice, which would mean it would alter the metabolism of the actual drug. An example of a drug that uses CYP3A4 are some of your heart medications, for example. Now, CYP2D6 is going to be the P450 that metabolizes codeine. And then we also have CYP2A1, which is a P450 that is going to interact with a toxin from a fungus. So you have a fungus, and this is Aspergillus flavus. And Aspergillus is everywhere in our environment, and Aspergillus is not going to impact you if you are immune competent. It's just when you get into hospital environments where you have a lot of immune compromised individuals, Aspergillus can cause a problem. Aspergillus flavus is also going to be growing on peanuts, and it's thought that that is one of the main causes for peanut allergies, is the toxin that is actually growing on peanuts. So aflatoxin B1 is made by Aspergillus flavus. And it's going to release this compound, and it's going to be metabolized by our CYP2A1, and it's going to be made more toxic. So in this case, our P450 is not protecting us, it's going to make it more toxic. And so this is going to be involved in hepatocarcinogenesis, and this introduces mutations into one of our key cell cycle regulators, P53. So basically, you have activation from your aflatoxin B1 into an 8,9 epoxide of the P450s, and the epoxide is modifying your DNA by forming adducts with guanine residues. And then CYP2E1 is going to metabolize ethanol, as well as other alcohols, and is going to metabolize acetaminophen. Now, acetaminophen is Tylenol, basically. And Tylenol is going to be metabolized by the liver and is going to be safely excreted. However, it's taken in at high doses, it can be toxic. Tylenol is going to be gluconated or sulfonated for excretion. So you can add gluconate, and then it'll go to the urine and kidneys. And you can also add a sulfate, and it'll go to the urine or kidneys. And then you've dealt with acetaminophen. Now, of course, acetaminophen is going to use CYP2E1, and ethyl alcohol will also use CYP2E1. In the presence of high concentrations of acetaminophen, or in the presence of alcohol, the CYP2E1 levels increase. And so it'll push the metabolism of your acetaminophen in this direction here. So this direction is where you don't want to go, but when you have high levels of CYP2E1, you're going to push it into that direction. So in the presence of excess acetaminophen, in the presence of alcohol, your cytochrome P450 is going to produce the toxic intermediate and acetyl P-benzoquinonamine, or NAPQI. Let's get, yeah. And so NAPQI is toxic. Now, NAPQI can also be made more soluble. Let me do this in green. Like glutathione. So you have the ability to make it more soluble, and then you're going to get a mercaptouric acid, which can be excreted in the urine. Or it can be toxic and affect cell proteins. Now, NAPQI is going to be dangerous, and it's also going to be unstable, and it can eventually lead to death of the hepatocytes. So that's why acetaminophen overdoses are critical. So your NAPQI is going to cause death of your hepatocyte. And under normal conditions, you have less than 10% of Tylenol that forms this NAPQI, and this can easily be handled by the glutathione detoxifying system. So this would be a phase 2. But when you have chronic abuse of alcohol, which this will lead you to have more of an increased sensitivity to acetaminophen toxicity because you have an increase of CYP2E1 since you're making more of it to deal with alcohol, then it pushes more towards NAPQI, and that overrides your glutathione. So treatments for acetaminophen poisoning are going to involve the use of N-acetylcysteine. I have it right here. And N-acetylcysteine is going to promote the formation of a less toxic compound, the myricapturic acid. Okay, and so another important medically important detoxing is with vinyl chloride. So vinyl chloride is used in the synthesis of plastics. So it's for synthesis of plastics. And it can cause angiosarcoma in the liver. Of people who work with vinyl chloride. So it's going to be activated in phase 1 reactions to be a reactive epoxide. So again, you have vinyl chloride here, and you get your phase 1. It's going to be that CYP2E1 again, and you're going to get a chloroethylene oxide. And this is going to be able to react with guanine in DNA or other molecules. So you can get binding to DNA and cell damage. It can also be converted to a chloroacetic aldehyde, and this can be conjugated with glutathione and excreted. So you basically want it to go in the direction of your glutathione, of course, to make it more soluble, but in the presence of excess, you're going to push it more towards the chloroethylene oxide and not be able to push it through the phase 2 reactions fast enough, and it's going to cause damage to your hepatocytes. All right, so moving on to some things that sound familiar. Regulation or major functions, of course, are going to be regulation of blood glucose. So you are going to have input from hormones to help regulate the liver and its metabolism. So as we all know, the liver is going to maintain blood glucose levels during fasting. You have growth hormone, cortisol, as well as catecholamine secretion that help to sustain normal blood glucose levels during fasting. And so you're going to have two key enzymes involved in this. You have, of course, cyclic AMP, and this is going to activate via the protein kinase, as well as the AMP activated protein kinase. Now, intermediate need for glucose, of course, glucose 6-phosphate is going to be dephosphorylated by the phosphatase in your liver. Break that again. And that will release, of course, glucose. And then you can also, if there's no need for blood glucose, of course, your glucose can form glycogen, NADPH, through the pentose phosphate pathway. Different nucleotides, cholesterol, fatty acid, triacylglycerol, phospholipids. So the liver is, of course, going to make all of those things. So you're also going to have metabolism homeostasis. Your liver is going to be important in the synthesis and export of cholesterol. It's also going to be important in the formation of triacylglycerols. The donation of nitrogen to the urea cycle. You are going to have ketone bodies made. And keep in mind the liver can make ketone bodies, but it cannot use ketone bodies. So the liver can use most things for energy. It cannot use the branched chain fatty acids, and it cannot branched chain amino acids, and it cannot use ketone bodies. And it also is going to be important for nucleotide synthesis. Now, you're also going to synthesize blood proteins. So the liver is going to be the key or the primary site of making proteins that are going to be circulating in the blood, especially serum albumin. And clotting factors. So when you have the liver protein synthesis compromised, so if you have a condition of hypoproteinemia, this could lead to edema because of a decrease in protein-mediated osmotic pressure in the blood. And this is going to cause fluid retention in the tissues. So this is going to be oncotic pressure, colloid osmotic pressure. Okay, so most abundant plasma protein is going to be liver albumin. This is going to be about 55 to 60% of your total protein pool. In the blood. And so this is going to be, as I mentioned many times, a carrier for fatty acids, for different hydrophobic amino acids, your steroids, hormones, vitamins, especially lipid soluble, and also just vitamins in general, as well as certain drugs. So a lot of pharmacological agents are designed so that serum albumin will pick them up because it'll help the half-life of the drug, for example. And albumin is also an important osmotic regulator. Other proteins from the liver are going to be glycoproteins. They are going to be involved in homeostasis as well, transport. They do a lot of protease inhibition. They protect proteins from degradation. And they're also going to stimulate hormone release. So they're going to be secretagogues for hormone release. So hypoproteinemia, which we're talking about here, is when your protein levels are less than 6 grams per deciliter. This can cause through sunburns, for example, through skin, through burns, through nephrotic syndrome, malabsorption. So causes are burns, malabsorption, malnutrition, nephrotic syndrome. So you have a loss of proteins through the urine, increased urine secretion of proteins, or decreased synthesis. So you have a liver disease. Now the liver is also going to be important for the synthesis of proteoglycans. Liver is going to need a high amount of mannose, fructose, galactose, and other amino acids. And they're going to make various glycoproteins and proteoglycans. So it's going to need mannose, it's going to need galactose, fructose. And so all of these are important in making all these compounds that we've talked about before. Now, another important function of the liver is glycosylation. So most of the sugars secreted by the liver are going to be O-linked. And so, again, we've already talked about O-linked, in which the carbohydrate is going to be attached to a protein at the anomeric carbon. So again, here's our anomeric carbon. And this is going to be a glucosidic link to the OH of a serine or threonine residue. So here we have a serine residue, and this could be or threonine. The N-linked arrangement is an N-glycosyl linkage to asparagine. shown here. And this will be N-linked, and this is O-linked. So an important O-linked sugar is an acetyluramic acid, or inanna sialic acid. And so this is an important O-linked protein that is going to be made and released by the liver. Now, another major function is in the generation of NADPH. Plus your 5-carbon sugars. And again, we've talked about all cells, including the red blood cell, have this pathway, and the red blood cell is referred to as the hexose monophosphate shunt. And it's because all cells need to produce NADPH. Your liver has a high need, similar to red blood cells, for NADPH so that you can keep glutathione reduced. And this is going to be important in protecting the membrane, especially from oxidative damage. This is also going to be important for the 5-carbon sugars. So you're going to make ribose. And then this is going to help, of course, to make your purines, pyrimidines, etc. The liver is going to have a very high demand for NADPH because not only is it going to be needed for oxidizing glutathione, but you also need NADPH for the synthesis of fatty acids, cholesterols, the synthesis of very low density lipoproteins, and biosalts. And so there are a lot of needs for this. You also have NADPH, which is going to be used by your cytochrome P450s. And so for all your detoxing, you also need NADPH. So your liver does need a lot of NADPH. So overall, the liver is going to participate in all of these reactions listed here, and they're going to use a lot of glutathione and a lot of NADPH just to carry out their normal functions and to keep the cell so it doesn't get damaged too much by alcohol or oxygen species. So we talked a lot about fuels used for the liver. The liver is going to need a lot of energy, so it has high fuel needs. And so it's going to consume about 20% of the total oxygen used by the body. And so, as we know, after eating a mixed meal where you have carbs, then your major fuels used by the liver are going to be glucose, galactose, and fructose. And then the liver also is going to be the major site of ethanol oxidation, and this is going to make acetate, which can then, of course, become acetyl-CoA. And then after an overnight fast, your liver is going to primarily or only use fatty acids. Your alpha keto acids that are going to be derived from degradation of amino acids are also going to be a potential source of fuel for your liver as well. So overall, with respect to fatty acid metabolism, again, this is all going to depend on what state that you're in, what the needs are from your liver, whether you need to generate bile salts, whether you need to make cholesterol nuclei for bile salts or steroid hormones, etc. So your fats can be converted to liver lipids. They are going to be, during the fed state, especially oxidized to acetyl-CoA and NADH. So fats can be, liver lipids, which is needed for their cell membranes, etc. Can be oxidized, and this is going to be acetyl-CoA and NADH. It can also, this will of course go to TCA, it'll also generate ATP, and excess acetyl-CoA can be converted to acetoacetate and beta-hydroxybutyrate. These are ketone bodies, and excess acetyl-CoA can also be used to form cholesterol. The fatty acids are going to be formed so they can make fatty acids, or they can use fatty acids to make tags. You can make phospholipids, different lipoproteins, and then free fatty acids can be bound to serum albumin and used by the heart and muscle cells for beta oxidation as well. Now, with respect to medium chain fatty acid, liver and kidney are the major sites of oxidizing medium chain fatty acids. So when you eat a mixed meal, your medium chain fatty acids are going to be released directly into the portal circulation, and they're going to be activated to your acetyl-CoA derivatives using the medium chain fatty acid activating enzyme, which is only present in the liver and kidney. So the kidney and liver have this. Now, of course, peroxisomes are going to be in every cell, but they're going to be in higher number in the liver than in other tissues. They are going to have about 50 enzymes associated with them. Of course, your peroxisomes are going to carry out your beta oxidation. Of your very long chain fatty acids, as well as your branch chain fatty acids. As we know, they are also going to have catalase. And catalase, of course, is going to be important for detoxifying hydrogen peroxide and converting that into oxygen and water. You have Zellweger syndrome. And this is an inherited absence of peroxisomes in all tissues. And so you're going to get long-chain fatty acid synthesis, which is necessary for myelin formation, is going to be impacted. Now, peroxisomes are going to have peroxisome proliferator activated receptors. This is shown here. Actually, I should just, these receptors here. So these are your peroxisome proliferator. And they got their name because it was found that if you add certain agonists, these agonists were able to induce the proliferation of peroxisomes. So you have, for example, shown here, you have your diet, adipose tissue that release fatty acids, and these fatty acids have a peroxisome proliferator activated receptor, and this will go on to turn on certain genes. So they are nuclear receptors that then become transcription factors, and they promote the proliferation of your peroxisomes. So there are three major peroxisome proliferator activator receptors. You have your alpha, which is the major form in the liver. And this is going to regulate activities of genes that are involved in fatty acid uptake and the beta and omega oxidation of fatty acids. And then the two others are the delta, beta, and then the gamma. Now, the liver is going to, again, be important for detoxifying xenobiotics. And so it's going to sometimes use pathways of fatty acid metabolism to detoxify, especially xenobiotics that are very hydrophobic and lipid soluble. So one example is going to be benzoate, shown down here. Now this is going to be naturally present in plant foods, and it's also added to sodas as a preservative. And its structure is going to be similar to salicylic acid, and that's a form derived from aspirin. So you can see here that here's salicylic acid. And so you're going to have a very similar process of making benzoate soluble as hippuric acid as you would aspirin. So this way to get rid of these fatty acids is very similar to medium chain fatty acids. You're going to have activation by the medium chain length fatty acid activating enzyme. And then it gets conjugated with glycine. So this is the step 2. Oops. And this is going to be the step 1. And then the glycine derivatives are going to be sialoglutarate or hippurate. And of course, one is for aspirin and the other is for benzoate. Now, when you have liver diseases, a chronic parenchymal liver disease, you're going to get changes in plasma lipids as well as lipoproteins. And some of these are going to be related to a reduction of lecithin cholesterol acyltransferase, or LCAT. So that's here. So in liver disease, you can get a decrease in LCAT. And this is because the enzyme is synthesized and glycosylated in the liver, and it produces the cholesterol ester as well as lysolecithin. Okay, so again, the liver is important, and so in this case, it's makes the cholesterol nucleus, and then combining that with the phosphatidylcholine or lecithin, you are going to get your cholesterol ester and lysolecithin, and that's via the LCAT, and then your LCAT is unable to function because it's decreased via the liver. So I should, oops, that kind of looks like the same. Decreases LCAT. So again, your levels of cholesterol ester are reduced. Free cholesterol in this case can be elevated. It goes back to all of our movement of fats. Your triacylglycerols are normally cleared by low density lipoproteins, and the activities of low density lipoproteins and lipoprotein lipase, as well as hepatic triglycerol lipase, are reduced in patients that have liver disease. So So lipoprotein lipase activity is decreased, as well as our hepatic triglyceride lipase activity. So that'll also be decreased. And so in people with liver disease or parenchymal liver disease, you're going to have high levels of triglycerides in the plasma. Um, you're also going to have impaired VLDL production. And so that is going to also impact, of course, the movement of cholesterol and lipids throughout the body. Now, amino acids are going to be important because the liver is going to make a lot of proteins. So the liver not only has to make its own proteins and enzymes, but it also has to maintain plasma proteins. And so your liver is also going to release amino acids. Oops. And these can be picked up by other tissues to use. It's also going to use amino acids to make nucleotides, hormones. It's also going to degrade amino acids. And these are the transamination reactions or deamination reactions to our different intermediates of your TCA cycle. And it's also going to convert ammonia to urea. It also can convert pyruvate to glucose. And then pyruvate can also be converted to acetyl-CoA, ketone bodies, etc. Your amino acids are going to put, the carbons of amino acids can enter TCA cycle, go into the electron transport chain. The carbons can also be used to form tags. And then, of course, your amino carbons going to TCA cycle intermediates can be used to synthesize glucose. And, of course, those are both through gluconeogenesis. Okay, so with respect to amino acids, if you have a person with liver disease, the concentration of amino acids in the blood is typically elevated. And this is caused by an increased rate of protein turnover, as well as an impaired ability of the liver to take up amino acids. The muscle is going to be a major source of the increase of amino acids, and this is seen typically in many catabolic states, such as cirrhosis of the liver. So with respect to cirrhosis, your plasma amino acids are going to show an elevation of aromatic amino acids, phenylalanine. We have cirrhosis down there. Let me write it up here, though. Your aromatics, phenylalanine, and tyrosine. And you're also going to have your free tryptophan as well as methionine. However, it's unknown why, but you also do see a reduction in the plasma levels of your branched chain amino acids in cirrhosis, but it's not known why. Okay, so when you have diseases of the liver, you're going to get elevated level of enzymes that are involved in amino acid metabolism, and those are your alanine aminotransferase, or ALT, as well as your aspartate aminotransferase, AST. So these levels will be elevated in the serum. This can be due to your hepatocytes being injured due to an infection, and then they're going to release these enzymes into the blood, and in the blood you'll see these increases. Jaundice is going to be an accumulation of bilirubin in the blood. And this is usually caused by inefficient bilirubin glucuronation in the liver. And this is going to increase clotting times. Which is caused by the fact that the liver just has a difficulty in making factors for secretion. Edema is going to lead to a reduction in serum albumin synthesis. And of course, that's going to have a lot of impact on osmotic regularities and things like that. There is also hepatic encephalopathy, which has reduced urea cycle activity. And this leads to increases in ammonia and other toxic compounds in the central nervous system. And then, of course, we also have our fatty liver disease, which is reversible, and then we have hepatitis 2 cirrhosis, and cirrhosis is, of course, non-reversible. Okay, and that is that.